Welcome. In this session, I'm going to continue discussing about molecular diagnosis. In the last session, I told you that conventional diagnosis methods will diagnose a particular disease only when the patient shows a particular concentration of the pathogen in his body. So here, of course, that is the time when the patient shows symptoms, and by the time the patient shows symptoms, the number of that pathogen or the concentration of that pathogen is already high. Now biotechnology is promising that a particular disease can be identified in a very uh, early in its stages. So the moment a particular pathogen enters into your body, that can be identified immediately by using these molecular diagnosis techniques, right? So like for example, in case of HIV, so it can be detected in very early stages. And I hope you remember from the chapter human health and diseases, so cancers, if they are detected in the early stages, and they can be completely curable, right? Now in the same way, if we are able to identify diseases in their early stages, the treatment becomes very easy. So that's what the biotechnological techniques like this rDNA technology, the PCR and Elisa test will help us to identify a particular disease in its early stages. So now, in the last session, I already discussed about recombinant DNA technology and PCR. Uh, okay, how they are used. So how they are used to find out the diseases. So even we have seen that the PCR is being routinely used even to detect the HIV, and it is also used to detect, okay, mutations in the genes of suspected cancer patients, right? So when we see this PCR, this is used in detecting HIV, and it is also used in detecting mutations, in, okay, genes of suspected cancer patients. So how we detect those mutations, so that I already discussed in the last session. So where you will be taking a single stranded DNA or RNA that will be radioactively labeled, and that is called probe. And the probe will have the normal sequence of that particular gene. It is not mutated. The original gene sequence will be present. And when you take the DNA of that, suspected cancer patients, and if there is a mutation, okay, say GC, so this is the normal sequence of a gene, say suppose is a normal sequence of a gene, and this is either single stranded DNA or single stranded RNA I'm going to take. And I'm going to take even the radioactively labeled DNA or RNA molecule. So this is radioactively labeled DNA or RNA molecule. Of course, this will be also a single stranded. It will be not a double stranded. So you need to take either single stranded DNA or the single stranded RNA. And usually this radioactively labeled single stranded DNA or single stranded RNA molecule will have, okay, sequence. So same like this. So G, C, I'm sorry, the complementary, there will be complementary sequences for this: C, G, T, A, CGGTAC is the complementary sequence that you will be seeing, okay, to that particular gene here. So this is a gene which we want to find out whether this is mutated or not mutated. If it is having mutation, and if we find that it is having mutation, then the person is having cancer. If not, the person is normal, right? So here, say suppose, if this is normal, so normal gene, it's not mutated. So when, if the gene is normal, then this complementary strand, that is single stranded DNA or whatever, you can take the RNA, but if you take here single stranded DNA, then you have to take single stranded DNA that is radioactively labeled. If you take single stranded RNA, then you have to take single stranded RNA that is radioactively labeled. Okay, so this is the probe. The radioactively labeled DNA or RNA molecule is probe, right? So this is how you will be taking the DNA. This is the gene that is normal gene, say suppose. If it is the case, then this will bind here. So it will get, okay, bind to this T A C G G T A C. So it will bind and it results in the formation of either double-stranded, double-stranded DNA or double-stranded RNA. And if it is the case, then this can be seen on a photographic film. So when you expose, once you allow this probe to become the complementary for this single stranded DNA or RNA, once it gets attached, this can be seen on the photographic film after the radiography. Right, now the point is that if this gene is mutated, so here now this is not a normal gene, it is a mutated gene. Say suppose somewhere here it is mutated to T. So earlier it was, okay, G here, so that was mutated. Now in that case, what happens is this complementary strand that is radioactively labeled will not be able to bind. So it will not bind to this, because, so you can see at this point, okay, it's a single base mutation has taken place, and as a result this will not bind, and if it did not bind, then it will be not shown on the photographic film. So that's what you have to keep in mind. The mutated genes are not shown on photographic film. The mutated genes are not shown on photographic film, right? Whereas normal genes, the normal genes are shown on photographic film. They are shown on photographic film, right? The mutated genes will not be seen on the photographic film. So just like how you see the X-ray films, so you might be knowing, so when you broke your hand, leg, and they might have taken that photographic film, so you can see that where the bone is broken and everything, you can see clearly. The same, the film will be used here to find out whether a particular gene is mutated or not. So because it is the complementary DNA or RNA is radioactively labeled, so when exposed to a photographic film, so it will be shown up on that photographic film. If there is no radioactively labeled, of course, when it do not bind to this DNA, of course, when it is mutated. So when this gene is mutated, this probe will not bind. If it is not mutated, then this probe will bind. and that will be shown on the photographic film, right? So this is how we are using the PCR, okay, in detecting the mutations in genes of suspected cancer patients, right? Then now we will see about ELISA. Okay, so this ELISA is used to detect either antigens or antibodies. So another one under this molecular diagnosis is Elisa test. So this stands for enzyme linked immunosorbent assay. So the full form of ELISA is enzyme linked immunosorbent assay, right? And this ELISA is used to detect antigens, or antibodies produced against, okay, that antigen. I hope by this time you might have understood from the chapter Human Health and Diseases that a particular antibody is specific to that antigen only, right? So if antibodies are produced, say suppose to antigen X, so antigen X is there in your body, right? And now an antibody is produced against this, right, antigen X. So that's antibody, which I say anti-X antibody. I'll just say anti-X antibody. It means that this anti-X antibody will detect only this antigen X. If antigen Y is present or antigen Z, no, it will not at all detect. It never bothers about that. Okay, so that's what we say antibodies are specific. So very much specific. Clear? So anti bodies are specific here, and a particular bacteria or virus enters, and they will be having antigens, and okay, so antibodies against that particular antigen of that bacteria or virus or any other pathogen will be produced, and they remain in the body, okay, for a long time, right, providing immunity to the person. Now, this ELISA can be used to detect that particular antigen. Say, suppose a bacteria entered into my body. Even in the low concentrations and whatever, you can find out whether that particular antigen is present in my blood, or the antibodies produced against that particular antigen, detecting either directly that antigen or indirectly the antibody produced against that antigen. So in both ways you can find out what kind of disease a person is having, okay, in its early stages, right? So here there are different types of ELISAs, which I'm not going to discuss: direct ELISA, indirect ELISA, and so here I'll just discuss one sample, okay, under this one. So I'm going to discuss about the sandwich ELISA. Okay, sandwich ELISA. This I'm discussing only for the purpose of understanding that what actually is ELISA, how they are going to perform this, right? Now, so first what they are going to do is that they are going to take okay, absorbents. So this is an absorbent material to which they will first attach antibodies. So this antibody is what we call it a captured antibody. It's called captured antibody. Right, so see, this is not in your PUC, just he has given a small paragraph, right, Only for the sake of understanding, I am giving an example. So if you understand it, so very well and good. Okay, right. So this is the adsorbent, and to that you are attaching a captured antibody. This antibody is specific to that particular antigen. Now what I'm going to do is, once I take these captured antibodies, I'm going to add that serum, okay, whatever the fluids which I'm going to collect, okay, from the patient, right, urine or whatever. And in that, if the antigens in fact are present, that antigen will bind. If antigen is present, then only it will bind. Okay, so this is the antigen that binds to this capture antibody. Not any antigen it binds, because I told you that this antibody is specific. Only, okay, that antigen, just like say here I will take that antigen X, then this antibody is anti-X antibody. So this is anti-X antibody, means this is going to find only antigen X. If you are adding antigen Y, it will not at all detect. Then I'm going to take another antibody. I'm going to take another antibody that is, okay, linked with enzyme. So this is enzyme linked antibody. So I have taken enzyme linked antibody, right? So this is the antibody and this is the enzyme. So what is this enzyme and all, that we will discuss okay, in the NEET class, right? So I'm just giving you a elementary idea that how this ELISA works. Now this is enzyme linked antibody here. Okay, so to the antibody we have linked an enzyme, and this enzyme will catalyze a reaction. Now the point is that this antibody will be specific to this antigen. If this antigen is present, then this will bind, otherwise it will not bind. So in this case it is present, antigen is present. Now you can see that the antibody, when added, okay, so that antibody which is enzyme linked, when it is added, it binds to that antigen X, and it has, the enzyme linked to this. So once this is finished, I'm going to add a chromogenic substance. So a chromogenic substance that turns into a particular color, okay, because the enzyme is present here. So this enzyme is able to catalyze the reaction, right, and convert the color. So here I'm going to add a chromogenic substance. So chromogenic substance, that is a color producing substance, okay, and a particular color is produced. Now if color is changed, then it indicates antigen is present. And if the color doesn't change, so when does the color change? When this antibody is present, which is linked with the enzyme. And when is this antibody present? When antigen is present. Say suppose if this antigen is not present, then this antibody will not bind, because there is no antigen. And after that, if I add chromogenic substance, then there won't be any change in the color. And if there is no change in the color, then antigen is absent and the test comes negative. And if that antigen is present, then okay, the antibody will bind, and now the antibody with the enzyme is present. And if you add the chromogenic substance, definitely there will be change in color, and that confirms, okay, the presence of antigen, and it's a positive, right? So this is how the enzyme linked immunosorbent assay is formed. So just I gave an elementary idea. I have taken a sandwich Elisa test that makes you to easily understand how the Elisa is performed. Right, so that's all about the molecular diagnosis. So the molecular diagnosis will help, okay, to detect the diseases in the early stages or in its initial stages, and by detecting the diseases in the initial stages will make treatment easy. Next one we shall discuss is about transgenic animals. Transgenic animals. Remember that I'm discussing this under biotechnological applications in medicine. So I'm still discussing the biotechnological applications in medicine, and under that the next topic is transgenic animals. So what these transgenic animals are? So whatever that animals in whom the DNA is altered by introducing a foreign DNA. Okay, so I have created an animal, okay, by introducing some genes that I have taken from bacteria or some fungi or some plant or whatever. I have inserted that gene into the animal DNA, and that animal is called, okay, transgenic animal. Like this we have produced transgenic mice, transgenic cows, okay, many animals into whom we have actually inserted these DNA. What I mean is a foreign DNA, right? Fine. So now let's look at first what's these transgenic animals. So animals, okay, whose DNA is manipulated. So animals, okay, whose DNA is manipulated. So means that is altered, right? And made to express a foreign DNA. So animals whose DNA is manipulated and made to express a foreign DNA. So just like when we see here the DNA molecule of that animal. So this is the DNA molecule of animal. And now what I did is I have introduced a foreign DNA into this. So you can see here we have introduced that foreign DNA. So this is the foreign DNA, and this is some animals DNA, right? And this is what I call it a manipulated DNA or altered DNA of that animal, and now the animal is called transgenic animals. And like this I told you already, we have produced so transgenic animals. So many animals DNAs we have altered, like we have made transgenic rats. So at present already we have, so it's not like going to make, already we have done it. So transgenic rats we have generated, then transgenic rabbits we have generated, and transgenic pigs, then sheeps, okay, and then cows, then fishes, right? So some fishes whose DNA is altered in that case, okay. So all etc are some examples here. So there are many others also, and scientists are not stopping. So they are continuously working on by introducing the foreign DNAs and see that how they are working, whether they are getting expressed or not getting expressed, and if not getting expressed, what could be the reason for that, and constantly they are doing research, right? So already we have produced some of these transgenic animals, the rats, rabbits, pigs, sheep, cows, fishes, etc, right? And here, out of all these, transgenic animals, right, over 95% of all these transgenic animals that exist at present, okay, they are all mice. So over 95% or more than 95% of transgenic animals are of mice, right? So we made a lot many, mice which are transgenic, so which will be used in laboratories. Many scientists are trying to find out the treatments for cancer. They take out the cancer genes, isolate them, and then put them into the DNA, okay, of these mice, and they grow these mice and see that, okay, how the mice is showing that cancer, and they will be given drugs, and how the mice is able to cope up with the drugs, and so on. So there is so many, varieties of research is going all around the world, and mostly they do it first on these mice, okay. So hence more than 95%, so not rabbits, pigs, cows and all. So because the mice are very easy to maintain in the laboratory conditions, and in small places also you can maintain them very well. But in case of these sheep, cows, the pigs, you require a lot of space, and you need to allot lot of space, and also the feeding and then taking care of them, and all that will be very costly in fact. So hence we are using mostly mice. Of course, once if it gets succeeded in a particular drug or something, they will be slowly moving on to test it on some higher animals like monkeys, okay, and then the close relatives of humans, and then finally if everything goes right, and they will be using directly on some selected human beings, and if everything goes right, then they are going to release. So especially like in case of vaccines, so once we generate a particular vaccine, okay, even now I think you may be knowing in this case COVID. So all over the world many are doing these researches, and on what they are doing the researches, usually on this like the transgenic mice, and so the first test on them, and then slowly they go on to the higher group of animals. And so you may be hearing about first trial, second trial, that is all the clinical trials and all. So it's the time almost to release those vaccines before that. They need to see that whether they are working perfectly or not, otherwise they will be not released into the market, and they have to start the research all over again, right? So now, in what way these transgenic animals are used in medicine? So let's look at the significance of these transgenic animals in medicine. So significance of transgenic animals in medicine, okay. So the first one is normal, I mean, to study normal physiology and development, to study normal physiology and development. I will discuss that in detail about this too, how they will be studying this normal development, okay, normal physiology, right, by using these transgenic animals. And these transgenic animals are also used to study diseases. So how that particular disease is caused. So if it is the case, if we come to know it, then we may get a solution to treat that particular disease. And then, right, so in production of biological products, okay, then, right, even in testing vaccine safety. In testing vaccine safety, so whenever we make a vaccine, and that vaccine need to be tested before it is released into the market. Otherwise, if it goes wrong, it can result in the death of many humans here. And also used in chemical safety testing. Chemical safety testing, right? So they are used to study the normal physiology and development, to study diseases, and used in making biological products, and in testing vaccine safeties, and in testing, okay, chemicals also. So for all this, we are using these transgenic animals, right? With this, I will end today's session. In the next session, I'll discuss each one of it in detail. So how they are going to use the transgenic animals, for what purpose, okay, and all that. So to study this normal physiology and development, all this we will individually, each topic, each heading, I'm going to discuss in the next session. Right.